Good evening, sir.
Good evening, ma'am. Good evening. Good evening, my dear students.
Good evening, sir.
Good evening,
Good
Pa. Good evening, Rotika and Varsa. The other students also.
You can start now, sir.
Yes, ma'am. Thank you. Thank you so much. Okay. So, good evening, my dear students. You are all are warm. Welcome to the wonderful session of the biological classes. Today, what we are going to.
Sir.
Good evening, Kaba. So today, what we are going to discuss about, so already we discussed what is meant by Mendelian disorder, isn't it? So what are the things already we studied, and hemophilia, phenylketonuria, and also other things also. See the question here, which mutation in beta globin? Note it down, my dear students. Hemoglobin, it contains two portions, is that? Two alpha chain as well as two beta chain is there. Particularly, two beta chain, what is that? Responsible for the abnormalities, isn't it? Already we studied the which position, that things also that as a result, what happened? The double standard extremely sorry. So the double disc shaped RBC molecules converted into or modified their structure into the which one sickle cell shape. Okay, what are the responsible of nucleotide? Means our about. So G, which one Anybody else? CAG and CAG, isn't it? These two only. So this is a modification which plays already discussed the sixth position. Sixth position means just imagine that there is a nucleotide is there, alpha as well as the beta. The alpha cell as well as the beta cell is also there. Particularly the beta cell, it has a differences. It has so many amino acids denoted as a connected with the nucleotide. Okay, connected with the it's formed as a nucleotide already in our report. With the help of the bond is called as a peptide bond, already know it. Because globulin is a protein molecules. And each protein molecules, particularly protein molecules are made up of amino acid, each an amino acids connected with the peptide bond. One amino acids of the carboxyl end connect with the another amino acids of the which one amino group. Okay, see that one. I will show you small whiteboard then only. Okay, see here. Take an examples. Okay, this is a poly peptide chain like this. Imagine. Okay, there is an amino acid sequence. Amino acids already in our, but there are so many amino acids there. There are 20 amino acids also there. Can you see it, Pa? Okay, yeah. So amino acid, simply we may call as simply amino acid is there. It's connected with the another one. isn't it? Okay, so likewise. So one amino acid consists of 4 components. The carbon atom is center and also having the, what is that? Fully occupied position is that. One side there is a carboxylic group present in it. Okay, this one is carboxylic. Now opposite there is a, which group is the amino group. Okay, so this is also present in it. So it is connected with another amino acid, simply called as the same, like this, another amino acids. Okay, so here the OH group is the, okay, the opposite side, which one, the same structure is also, this one is a first amino acid, and this one, another one is a second one. Imagine this one is second. Okay, so the structure, same structure will be repeated here. Okay, so as a result, what happened? C is there, same structure is also present in here. Imagine NH2 here, and also opposite side as it is the COOH. Okay, how it will be the bond formation takes place, already told you, the first amino acids connected with the second amino acid with the help of bond is called as, which bond? This bond is called as peptide bond. How it will be? Peptide. Okay, peptide bond. Otherwise we may, what is that? We may put a text, it is better for you. Okay, peptide bond. Peptide bond is there. Where is it? Here in these places there is a peptide bond. How it will be formed? The acid group, there is a OH is there. The another amino acid, there is a amino group is there. These two are united together form as a, what is the peptide bond? Removal of the water molecules from this portion. What happened? There is a removal of one, what is that? Water molecules from this place to remove the 1 molecules of water. Note it down, my dear. Here the water molecule is completely removed. So there will be like this. Okay. So how it will be? See here, the H2 is here. You have to take the H2 in this portion, and also this O is also, you have to take this problem. So as a result, CO double bond NH only formation takes place, isn't it? See here. So here you have to take a oxygen molecules. Addition with this is the H2, isn't it? You take it out. So the water molecule, isn't it? Water molecule removed. Remaining is what happened here. What is the remaining portion is there? Is what is there? CO and also H is there, and also here there is a N only. The bond formation, how it will be formed? C and O double bond. What is that? N H only, isn't it? So in between there is a bond. This is the bond. What is that bond? That bond is called as, what is that? Peptide bond. Okay. Likewise, it's forming, particularly the changes occurs in which places? Already a little bit. Which place? The 6th place. 6th or 7th place. Which one, pa? 6th place or 7th place? The changes occurs.
6th place, sir.
Very good. 6th place only, they have the changes, isn't it? See that? So do you understand this one? How it will be formed? Okay. So why we are discussing this one? Because the first amino acid is 2 and 7 amino acids also there. The 6th position of the amino acids, it contain, what is that? Glutamic acid. Glutamic acid. Simply we may call as GA. GA is the amino acid. The glutamic acids replaced by which one? Replaced by valine. Valine. Okay. So that is the things. Valine. Okay. So you will get it the clear idea. So as a result, what happened? The double, what is that? Disc-shaped structure and also oval-shaped structure. It may be modified into which one? Double shape. Real body. That things also. This is considered as RBC. So there is a disc-shaped structure like that. Okay, this is RBC, normal one. RBC. Okay, so this is a normal RBC. Okay. This is turned into due to the modification of which one, this portion, glutamic acid, the sixth place modified into valine. What happened, the RBC nature, it will be changed. RBC shape also changed into how? So look like a sickle cell shape. This is the structures due to the modification. Okay, they understand that one, my dear. Okay, so okay, see that one. Okay. Yes. Yes. Okay. See, and just similar to that of the hemoglobin. Okay, the cystic fibrosis also is the cystic fibrosis. Already we discussed sickle cell anemia. Already we discussed this is a shape. Okay, and the portion glutamic acids. So it is also a term GAG. Okay, GAG is converted into how it will be. That is denoted with 3. GAG is what is that changed into what is that valine GUG. You got it down. So this is the changes occurs in the beta chain. So this is the changes GAG glutamic acid and GUG is the valine. Already we discussed that things in the last class, isn't it? This is the portions. So here there is a change, KH and CDG, like that it comes. Okay, go to the next one. So this is the same things. The sixth place is replaced. Okay, sixth position is replaced into another six. See, the mutant hemoglobin's molecules undergoes a polymerization under the low oxygen tension, causing the change. This is very, very important. Low oxygen change tension. Okay, suppose the low oxygen tension means what happened? It will not maintain their structure. Okay, so as a result, what happened? The RBC from biconvex disc to elongated sickle-like structures. So they cannot be able to carry the oxygen molecules properly from the one place to another place. As a result, what happened? We cannot be able to breathe properly. Okay. And the next one is already we discussed phenylketonuria. This one is also lack of the enzyme from the phenylalanine to into tyrosine. By the enzyme is called as what is that? See that phenylalanine hydroxylase. Hydroxylase is nothing but the OH ion is added, hydroxyl group. Okay, phenyl hydroxylase. So phenylalanine is the phenylalanine. How it will be obtained? From the dietary sources of the protein. Suppose if it is not provide the dietary sources, it will uptake from the tissue protein, myosin. Okay, and so many protein molecules also present in it. Particularly, tissue proteins also degraded. And it gives a amino acid. The amino acids were phenylalanine. The phenylalanine was taken after the degradation process. It entered to the which one so many pathways there. One is phenylalanine converted into tyrosine pathway. Phenylalanine also converted into phenylpyruvate. After the conversion of phenylpyruvate, it entered the lactate and also what is a phenyl lactate is also same things as that with different steps. Likewise, the phenylalanine is also converted into tyrosine by the enzyme is called as phenylalanine hydrazine. This enzyme is lack. What happened? The conversion is not possible. Okay, the conversion is not possible. What happened? The phenylalanine deposited in the blood. So deposition of blood, what happened? It will lead to some other difficulties. It cannot be filtered by the which one kidney. Okay, so it cannot filter by the kidney. Means what happened? It will be deposited in your blood. As a result, it lead to some symptoms. Okay, see that. So this is a normal biosynthetic pathway present in your liver portions. Where it occurs, the phenylalanine converted into tyrosine. The conversion takes place within the liver, hepatic tissues. See that hepatocytes. The hepatic tissues, particularly the cell is made up of what is a hepatocytes. Hepatic cell is called as hepatocytes. So what happened, the conversion diet protein molecules, the phenylalanine is also derived or obtained. It entered into the hepatocytic cells, liver. So what happened, the conversion is also converted into, conversion is tyrosine. This tyrosine, after that conversion, it will be entered to the another process, which occurs within the liver cells. Again, it will enter into the blood only. Okay, this is the normal process. Suppose here the absorption, here there is a lack of enzyme. What happened? It lead to diseases. Clinical symptoms also expressed. That is very, very important, my dear students. Okay, see here. The which enzyme is responsible for the conversion of phenylalanine into tyrosine? Okay, already we discussed. Tyrosinase.
Option D.
Yeah, very good. Phenylalanine hydroxylase. Okay, see that one tyrosinase, phenylalanine carboxylase. Similar to this one. See here, carboxylase, hydroxylase. Methionine and as a phenylalanine hydroxylase. You know very well about the answer before we studied. Okay, phenylalanine hydroxylase. Okay, so no doubt at all, isn't it? This is very, very important. Sometimes they may ask the question. You have to directly. Okay, the phenylalanine is the aromatic amino acids or normal amino acids. Phenylalanine, methionine, cysteine, cysteine. These are all aromatic amino acid. Aromatic means what is the meaning? The ring structure, the hexagonal ring structure is the hexagonal ring structure, just like a phenol group. Okay, this is very, very important. See the accumulation of this in brain, note it down. So it is not filtered by the kidney. These are also excreted through urine because of its poor absorption by kidney. Suppose the absorption is very less in nature. Suppose if it is not absorbed by the kidney, what happened? It accumulated in the brain cells. As a result, it lead to the mental retardations. Okay, this is very, very complicated. So when you are taking the fish or meat or whatever it may be, so you have to drink more water. Then only it can be elucidated from the kidney. Suppose if it is not there, what happened? It may be converted to some other form, urea, uric acid, creatine, creatinine, like that. As a result, what happened? It will lead to some clinical disorders. Okay, note it down. And question part, that's all about, I think, which one of the following inheritance takes place only male? Note it down, my dear students.
Sir, Hollandric inheritance.
Yes, andric. Okay, here nuclear, it is not possible. Cytoplasm, these are all, or what is that? And cell-bound organelles. Cell-bound organelles. Codominance means both the parental character expressed in the mean. That means male as well as a female. Okay, isn't it? So it will not come to the which one, the male part. So the whole andric inheritance is the correct one. Andric means male only. Okay, so this is very, very important one. And second things already we discussed. Which of the following genotype represented intersex Drosophila melanogaster? Okay, Drosophila. See the 1A and triple X is that. Triple X denoted as female. They are having extra female characteristics. And here 2A plus XXY. This is the female, but having the male. This one is the correct answer. And then remaining all are, what is that? It is not a correct one. We will discuss. Okay. And then what about the third one? Already we discussed. Which one of the following is the correct statement of the sickle cell anemia? Already we discussed glutamic acid by valine at the 6th position of the beta globulin chain of the hemoglobin molecules. Sometimes they may be confused. 6 and 16. Okay. So only you must note it on 6th place. 6th place. Don't forget, my dear. Before we studied. Okay. And next one, the single gene mapping is observed in the fungi. Penicillium is also one of the fungi. Meager is also, Neurosporo and yeast also. But Gene mapping aspect, it will not come, these three. Which one is blue? It will not come. Neurospora, mostly, what is that? Used for the gene mapping observation fungi. Penicillin. What is the main use of the penicillin, my dear students? Penicillin is also one of the fungi. Penicillium notatum, Penicillium chrysogenum. Okay. They used for the, what is that? Antibiotic productions. Penicillin. Okay. Who discovered penicillin? Alexander Fleming in the year 1928. These are all very, very important. Mucor, already in our bread. Okay. So that bread mold, it contains mucor. Neurospora, it's a very important one. It is also very, very important. Already we studied. Okay. Neurospora. So, and the last one is called as yeast. Compared to the other one, this is also very, very important one. Will you please tell me? These three, these four, among these four, the yeast is a specific one. Will you please, anybody? Do you have the ideas about the yeast? Varsa?
Yes, sir. It is used for fermentation.
Okay. One is I'm not asking about the fermentation. According to structure only, I'm asking. Any idea? Dhanalakshmi? You don't know, isn't it? Okay. Listen here. So all are innovative body except yeast. Except yeast. Which one is a multicellular? Which one is called as a unicellular? Means yeast is a unicellular fungi. Others are, what is that? Multicellular in nature. That is very, very important one. Okay. So this is a very, very important. And I think that which amino acid is changed question 6.1. Already in our about beta globulin protein. There what they asked, you know, the genetic aspect they are asked. Okay. Triplet codon aspect, they asked the question in that particular question. But in this category, which amino acid changed at position 6 in beta globulin protein causes sickle cell anemia? Here they are asking the amino acids aspects. See that glutamic acid replaced by valine. The answer is A only. Okay. Got it down? Okay. So that's all about the fifth part. We'll pass on to the next one is sixth part. Okay. See that one. Yes. Next, we will pass on to the next part is called as chromosomal disorder. Okay. What is my chromosome? What is the use of the chromosome? How many types of chromosome is there? How many pairs is there? Which one is allosome? Which one is a sex chromosomes? Already we know, isn't it? See here, chromosome is very, very important. They are possessive some specific, what is that? Characters, genes. Okay, they carries it. Before that, we must know what is meant by pleiady. Okay, what is by pleiady? What are the types of pleiady is there? How they are classified? That things we must note it down. So see here, what is by pleiady? The number of set of chromosome in a cell or organism is called as pleiady. Number of set of chromosomes. How many set of chromosomes we are having? How many set of chromosomes we are having? 23
Sets
Pairs.
of chromosomes.
Yes, we have to tell as a pair, isn't it? 23 means you are telling about the 20 plus 3. Oh, that is denoted as which one, you know, haploid nature you are telling, isn't it? You have to tell. 23 is also present in where it is. 23 is also present already. You told where it is present, whether it is somatic cell or sex chromosomes.
Sir, in the 22 pairs are allosomes and one is sex.
Okay, you are telling about that totally. And what I'm asking now, during the gamete formation, how many chromosomes are present? Dhanalakshmi?
23 only, sir.
Yeah, that's a good. That's a good. Okay, so when you are 23 chromosome is present in it, when you know after the what is a gamete formations? Already know about gamete formation. During the gamete formation, 46 chromosomes, the diploid nature produced a haploid gametes, 23 pair from the, 23 chromosome from the male gamete and 23 from the female. Both are haploid in nature. When it united together and fused together as a result of formation of zygote, that condition is called as what is a diploid 2n stage. N plus n, that is also addition only. N plus n, you will get 2n. Okay, we will see one by one. So you must note it down. Set of chromosome in a cell or an organism is called as ploidy. For example, haploid means one set. Diploid means two set of chromosomes. Already we know very well. Haploid is denoted as n and diploid is denoted as 2n. See here, the ploidy, another point of view. See that ploidy level is a term referring to the number of chromosome sets in somatic cell of the diplophase or gametophytic cell of the haplophase. Note it down, my dear. Somatic, gametophyte. Already I told you, gametophyte means haplophase because male it possesses 46, but in the case of gamete formation, 23 is present in it. That condition is called as haplophase. Okay. So diplophase means, you know about somatic cells, we are having 46 chromosomes. Okay. They are collectively called as what is it? Polyploidy. Okay. See here. Next. So this is called as haploid. See that only one chromosome is present in it. Only one. So this condition is called as haploid in nature. Suppose two chromosomes present in it, that is called as number of chromosome is 2, that is denoted as 2n. Why we are selecting these type? You know, this is a small one. This is the compared to the previous one is a slightly largest one. This one is a completely larger. There are 3 set of chromosomes we have taken. Each one it is a diploid nature. This one diploid, each one diploid in nature. But this in this category, triploid nature. Each and every chromosome they exist in 3. 2n plus n, that is called as triploid. N denoted as number of, what is that? Numbers. Simply, we may call as number. Here we are mentioning which one, you know, the chromosomes. Okay. So haploid and diploid and triploid and the tetraploid. Tetra means 4. So 2n plus 2n. Okay. Otherwise, here 2n plus 2 only they give. Okay. So these are all what is that? Types of see, they may be exist in 4 forms. We pass under them. So these chromosomes have some physiological changes occurs due to the mutation. Physical and chemical and biological factors it will affect. So as a result, what happened? No, the chromosomal, what is the nature also vary. The condition is also treated or inherited to the one generation to another generation. That condition is called as what is a chromosomal disorder. Okay. See here. So chromosomal disorder. What is meant by chromosomal disorder? See here. The cause due to the absence or excess or abnormal arrangement of one or more chromosomes. Okay. Already we saw it. Diploid nature already in our level. Triploid means addition of one chromosome, isn't it? It is a abnormalities. Suppose 2n plus 2, what happened, you know? 2 is added. So it is also another normal one, you know, diploid nature only. Suppose we are adding 1 or 2 or more than 1, condition is called as what is that? Chromosomal abnormalities. Okay. So it is lead to some disorders, diseases. See that. Another one, the failure of the segregation of the chromatid during cell division cycle result in the gain or loss of chromosome called as aneuploidy or heteroploidy. What happened here? What is meant by aneuploidy or heteroploidy? That means, what is that? Failure of the segregation of the chromatid during the cell division cycle. Okay. So as a result, what happened? It will not secrete from the one generation to another generation. During the time, what happened? You know, one or more chromosome increases. Okay. As a result, what happened? It will lead to some difficulties. Okay. That condition is called as what is it? Aneuploidy or heteroploidy. This is one point. And second one, failure of cytokinesis. What is meant cytokinesis? Cell division after telophase stage. Note it down, my dear students. Okay, telophase stage of cell division result in an increase in a whole set of chromosome in an organism, and this phenomenon is called as polyploidy. Where it occurs during the failure of cell divisions. Okay, particularly cytokinesis takes place is there. Cytokinesis means a single cell divided into two, isn't it? After telophase. Okay, after telophase stage, what happened? The cell division result in an increase in the whole set of chromosome. Within the cell, there is a chromosome is there. The chromosome also what happened? Increased. How it will be increases? See that one. That condition, that phenomenon is called as polyploidy. Ploidy, very both. Gain or loss. Poly means more than one. That is the mean. Okay. See that? So this is normally we are having the chromosomes. Human body, we are having these type of chromosomes. What is that? 1 to 23 pairs. Particularly 46 chromosomes are. Each one it has a, what is that? Diploid nature. 23 pairs. Up to this level, see the different types of structure is there. See that 1, 2, 3 are similar. Here up to the so sixth one, I think sixth one. These two are similar. See, these two are similar. Up to these two, which one Up to these three are similar. Okay. What about this one? This one is different. Okay. So this one is also different. These 12th one as well as 10 and 9, these are all similar. What about these three? See the structure? These are all similar in nature, iso nature. What about these three? So this one is different from these two. Okay. But these two are similar, but these two are similar. How many types of chromosome according to their name? These are all are considered, it is known as autosomes, somatic chromosomes. And the last one is called as X and Y chromosome is there. This is called as sex chromosome. Okay. This is called a sex chromosome. These only decide your, what is that? Male or female characteristic feature. Okay. This is the normal. They are all are monosomic. Okay. Suppose changes occurs in this portion, what happens? It will lead to some syndrome. We will discuss later. See here. After that, these are the things already we discussed. After that, we are going to discuss a chart. Types of chromosomal abnormalities have been stated, denoted as a small chart-like structure. What is my ploidy? How they are classified? There are two types have been reported. One is called as euploidy, another one is called as aneuploidy. Euploidy also further divided into monoploidy, diploidy, polyploidy. Okay, here the number of chromosome is whether increased. That is the reason. Again, the polyploidy also divided into two things is there. One is called as autopolyploidy and allopolyploidy. Automatically, the polyploidy nature is also occurs. Sometimes we are doing that's artificially. Okay, this is the one part. Aneuploidy, divided into two is hypopolyploidy. Hypoploidy means reduction reactions, reduced format. Hyper means increased format. Hyper, higher. Increase the number of set of chromosome, the particular gene or whatever it may be. Hypo means reduced. How they are classified? Hypo means reduced means monosomy. Monosomy means what is that? 2n normally present in it. That is called as diploid condition. When -1, what happens? You will get 1. So that is considered as a monosomy. Nullisomy. Nullisomy means what does it mean? Completely nil. 2n minus 2. What will be? It is related to 0 like that. So that condition is called as nullisomy. You have to note it down, my dear student. Monosomy. 2n minus 1. You will get 1. So that is called as monosomy. Nullisomy. 2n minus 2, that is nil, that is called as 0. That condition is called as nullisomy. Hyperploidy, trisomic, 2n plus 1 here added. Yeah, that is why, that's why it comes under the hyperploidy nature. Okay, 2n plus 1. Tetrasomy 2n plus 2. These are all what is that? Aneuploidy, but actually hyperploidy nature respectively. Okay, got it, my dear students. The same things is also given in the different format. Okay, here also the monosomy, nullisomy, trisomy, tetrasomy, pentasomy, hexasomy, penta, hexa, tetra. This is 4 and 6, 5 and 6. Okay, here hyperploidy, triploidy, tetraploidy, pentaploidy, and also hexaploidy. This is also same. Tri means 3, 4, 5, 6, 4 and 4, like that. Multiplication aspects. Okay, see this one. So this is a normal one, normal development. Diploidy into polyploidy. Okay, see how it will be happen. The single cell converted into endo replication. Inside there is a replication takes place. As a result, they will get it polyploidy. Here, see the continuous. Here, see the error happen. What happened? No, it may be divided into 2. This one is a depolyploidizations, error prone. During the cell division, some of the error also made due to the naturally or artificially. That time what happened? No, this is the normal one. Is that sometimes it happen like this. Okay, the aneuploidy and the copy number variations oncogenic genes. You know all about that. Oncogenic is nothing but one of the mutation, isn't it? The cell as well as the cell division also takes place due to the physical and chemical and radial, whatever it may be. That time what happened? The single cell divided into a number of cells. We cannot be able to calculate. Okay, it is due to the mutations and sudden changes. It is also see that before we studied. In which stage the cell division is failure to during polyploidy? Which stage? Anaphase, it will not come. Metaphase is not come. And diagnosis is not come. Which one you know?
Telophase.
Yes, good. Telophase only. Okay, so note it down, my failure, cell division failure during the polyploidy nature. Telophase is there. And aneuploidy, otherwise called as heteroploidy. See that aneuploidy and heteroploidy. What is aneuploidy? How it will happen? How they are classified? See that aneuploidy already we saw it in the chart aspects. We are going to discuss one by one and clearly. Aneuploidy or heteroploidy is a condition of the having fewer or extra chromosomes than the normal genome number of the species. It is called hyperploidy. When the loss of chromosome occurs and hyperploidy when addition of the chromosome occur. Heteroploidy or aneuploidy is nothing but what is the extra, fewer or extra chromosome than in the normal genome. Normal genome, suppose if you are having 2n is a normal one. Okay, 46 is a normal one in the human being. Suppose you are adding any one of the X chromosome or Y chromosome, what happened? 46 plus n, 47 it comes. Suppose 46 minus 1, what happened? 45 is come. Isn't it? So it will lead to some difficulties, some symptoms. That is called as, what is that? Aneuploidy. See, types of aneuploidy. See that monosomy. Okay. Another one is called as what is the nullisomy, trisomy, and tetrasomy. These are all what is that? Type of aneuploidy. Don't forget, my dear students. What is my aneuploidy? Whether it may be less or gain of chromosomes. Okay. As a result, what happened? No, it will lead to some difficulties. Okay. Suppose, take an example, XXY. What will be the nature of the person? Dhanalisimi? XXY.
Sir, male expressing female characters, sir.
Very good. Exactly correct. Okay. So, listen here. Likewise, there is a symptom. Okay. Monosomy. Monosomy is the loss of single chromosome represented as 2n minus 1. Already we discussed, isn't it? 2n minus 1, you will get n only. That condition is called as monosomy. Okay. See that? And nullisomy, already we discussed, is the loss of both member of the homologous pair of chromosome. It is represented 2n minus 2. Trisomy. Trisomy means addition, hyperploidy nature. Okay, here nullisomy means 0, just imagine. The gain of single chromosomes represented as 2n plus 1. Particularly, a human trisomic zygote has 47 chromosomes. Already told you, we discussed, isn't it? X, X, Y. Sometimes what happen, triple X is also present. Sometimes triple Y is also. It is very rare cases. Okay. And tetrasomy is the gain of two homologous chromosomes represented as 2n plus 2. Already we discussed. Okay. A human tetrasomic zygote has 48 chromosomes. A human tetrasomic zygote has 48 chromosomes. Already told, only we are having 46 only. Suppose we are adding the 2 chromosomes, means what is that? This is a 2n stage is 46. When you are adding 2 chromosomes, as a result, you will get 46. This is a, what is it? Tetrasomic zygote. Okay, how it will be possible, will you please see here? And you can understand, then only it will be better. Okay, see that one. See here, take an examples of male, okay? Male, and another one is called as female. How many chromosomes is there? Male is denoted as X and Y, isn't it? Here, X and X, isn't it? So what will be the heterozygous nature? It produces 2 types of gametes. One is called as X, another one is Y. Okay, here, about one is that only X chromosome, X gametes will be formed takes place. Here, how many chromosomes is there? Here, 46. Here also, 46. So during the gamete formation, what will happen? Here, only 23 is present, isn't it? 23 is present. See here 23. Here also 23. Likewise, here 23, isn't it? This is the normal. So after the fusion of gametes, sometimes it will not, what is that? Split like this, equal portion, 23, 23, it will not split. Sometimes it may split into how? 22, sometimes it is 24. Okay. So what happened here? So this is the male gamete, isn't it? The male gamete is also combined with the female one. What happened here? The 24 is the, here the 23 is the. So you'll get 40. How many is there? 47. So here, what happened? Imagine just XY, X, X is Y. The male having the female characteristics extra. That is very, very important. Suppose these two are united. So as a result, you will get it. How many things is there? 45. Isn't it? 45 only. So this is what happened here. One chromosome is lack here in this place. Here, one chromosome is added here. Okay. So these are all are very, very important things. Okay. Don't forget, my dear students. This is very, very important one. Okay. Each and every time I have to move to, there is a problem here. Okay, just listen here. So as a result, what happened, there is a changes in the chromosome and also lead to some other difficulties already we discussed. So here, so that's the things. This is the things, 48 chromosomes. Suppose if it is a tetrasomic, means the 2n is the 46. Suppose sometimes it may be 2 chromosomes is added the diploid nature, we will get it 48 chromosomes. So this is another thing. See that terms and chromosome numbers. Monosomy 2n minus 1. Double monosomy is also possible. Double monosomy. First division is also takes place. Second also 2n minus 1, again minus 1. It is also similar to that of the nullisomy, isn't it? 2n minus 2 only, like that. Okay, but it happened different divisions. And nullisomy is also there. 2n minus 2. Trisomy 2n plus 1. Tetrasomy 2n plus 2. Pentasomy 2n plus 3. Okay, each one it has a specific examples and also disease also there. So we are going to see one by one clearly. Okay, see that one. This is 2n stage. This is the first normal chromosome is that. This is a gametes. 23 chromosome, 23 chromosomes. You will get it the 46. This monosomy means what is that? 2n minus 1. See here. 1 is that one is 2n is there. Okay. It completely lack of 1. What happened is now? You will get it the only 1 is there. So this is monosomic. Trisomic means there is a diploid nature, the addition of 1 chromosome, so we will get it 3. So that is called as trisomic nature. So this one is also trisomic. See that? 2n plus 1. So we will get trisomic. Tetrasomic is here 2. Tetrasomic. Pentasomic means 3. Likewise, what happened? No, there is a changes is there. This is also another type of example. Euploidy, balance, and aneuploidy. Okay. See that? Nullisomy, completely lack. Disomy, double. Okay. Monosomy. This one is also single. 2n plus 1 trisomic. So see here, 2n plus 1, 3. The particular chromosome will be, what is that? Increased in this portion. Here, see that? High grade of aneuploidy. High grade means, what does that mean? Each one it is. Here the 2, here 2, here 2. See that these 3 is 3. Here also 5 is that. So these are all are high grade aneuploidy. Subchromosome. These are all are due to the some, what is it? Abnormal addition, deletion, substitution, inversion. Likewise, it's coming. Duplication is very important. Suppose these are all are comes under the duplication process only. Okay. This one is, what is that? So suppose if it is a diploid, 2 set of chromosome is there, 1 is lack. So this is called as deletion. This one is also deletion sometimes. Here, what happened? So 1 is 3 set of chromosome is there. Okay, in the pink color. Imagine. So this is also considered as a, what is that? Duplication. Okay, likewise. See here. And euploidy, change in a set of chromosome, they are divided into two. One is haploid, another one is called as polyploid. Haploidy, otherwise called as monoploidy. Haploid or monoploid, otherwise denoted as N. Only one set of chromosome present in it. Organism or hemizygous. Note it down, hemizygous. Okay. Normal in gametophyte, insect, rotifers, etc. These are all are normal in gametophyte. No meiosis, smaller size, less vigorous growth, leaves small, high sterility. Highly sterile in nature. Suppose XY chromosome is there. Okay. Suppose the Y is absent. What happened? That is called as highly sterile. Suppose XY, X0. Sometimes the X0 is also comes up. Okay. These are all are, what is that? Infertility. In females also you will get it. See that. Polyploidy. Polyploid in the presence of more than 2 set of chromosomes, already know about, most eukaryotic organisms are diploid. For the most of their life cycles possessing 2 set of chromosomes. Occasionally, whole set of chromosomes fail to separate in meiosis or mitosis, leading to the polyploidy. Already we discussed it. What is that? During the cell division, what happened? Failure of cell division, isn't cycles. So as a result, what happened? Leading to the polyploidy nature. So more than 2 genome set of chromosomes. This is polyploidy, include triploidy, tetraploidy, pentaploidy, even higher number of chromosome sets. Polyploidy is common in plant and a major mechanism by which new plant species have evolved. But there are so many varieties have been reported. For example, Brassica is one of the plant. Okay. Another one is also is there, cabbage. These two are crossed with it, you will get another new varieties. Okay, likewise, and Triticum vulgare, Triticum wheat. So these are the 2 things also crossed. As a result, you will get the variety. One is height, another one is dwarf. You will get the semi-dwarf. That means this one is 100% height, 100-meter height. This one is 40-meter height, like that, So in between, what happened now? 50-meter, you will get it. That is the new one. Approximately 40% of flowering plant species and from 70% to 80% of the grasses are polyploidy. Note it down, my dear students. Maximum number of, what is that? Grasses only the polyploidy nature. This is also another question. 40% of the flowering plant. That is also comes under the headings. Which one angiosperms. Flowering plant is also angiosperm. Gymnosperm is also comes, but it is not a flowering plant. Flowering plant, but it is not a naked one. Already know about. But in this case, 70 to 80% polyploidy. This is the NEET question, my dear students. Polyploidy is less common in animal, but it is found in some invertebrate, fish, salamander, frog. These are all are considered which animal sources. Animal is an example. Okay. So here you must know it. What is polyploidy? What are the types is there? How it will be? How many percentage present in plant? How many percentage present in animals also? Particularly 80% is grasses. Okay, this is the question already asked in the 2018-19. Just go and refer it. You will get you the ideas. Okay, see that haploid, only one chromosome is there. Diploid, both are diploid in nature. These are all 2n stage. Triploid, 3n, 3n, 3n. Tetraploid, 4, 4, 4. Hexaploid, each one it has 6 chromosomes. These are all are what is the nature of chromosomes. See that. And there are two types of major types of polyploidy: autopolyploidy and allopolyploidy. Autopolyploidy means, what is the meaning of autopolyploidy? In which all chromosome set are formed a single species, that is called as autopolyploidy. Or single genes only, or single species. Single species, what do you mean? Exit polyploidy, that condition is called as autopolyploidy. Allopolyploidy, what is the meaning? In which the chromosome set are formed two or more species. For example, take an examples of tomato. Okay, you will get the another new variety of tomato from the previous species. What is that? That is called as autopolyploidy. Allopolyploidy means, take an example, tomato and also potato. You will get pomato. That condition is called as, what is that? Allopolyploidy. Okay, you got it? See here, autopolyploidy. The autopolyploidy happens due to accident only. It's a natural one, so it happens accidentally. Or meiosis or mitosis, that produce the extra set of chromosomes, all derived from the single species. Okay. For example, a particular onion, particular tomato, particular grapes. Okay. Take an example, rice. So many things is there. Okay. MS Foundation, just go and refer it over how many new varieties are also introduced or produced or discovered. Okay, you can get it the answer. Okay, non-adjoining, non-disjunction of all chromosome in mitosis in an earlier 2n embryo, for example, double of chromosome number and produce a autotetraploidy. The 2n embryo also further what happened automatically what happened undergoes the divisions. Due to the mitotic division, you will get it the 4n stage. Okay, this is automatically happen inside the plant only. The same things is the autotriploidy is also there. Already I told when the chromosome is split, you will get 23, 23. 23 combined with the another one of the, it will not equally divided. Sometimes it may be unequally divided automatically. 22, 24. Okay, 22 always again join with any one of the male gametes or female gamete, like that. Imagine which one is female. Suppose one is female, consider means another one is a male. Okay, so the female division is 23 or 24, like that means the 23 join with the 24. As a result, what happened now? It may be excess. Sometimes it may be less number, maybe like this. Okay, see that. Alternative triploid may rise from the grasp between the autotetraploid that produce 2n gametes, diploid that produce 1n gametes. Because all the chromosome set or pro-polyploidy are formed the same species, they are homologous and attempts and to align the prophase 1 of meiosis. Note it down, my dear students, where it happens. Suppose if it is a triploid conditions, where it occurs, the attempts to align, it may become to the straight line. Okay, in prophase 1 of meiosis, because here what happened, in the sexual chromosome only we are having two types of prophase 1, prophase 2, isn't it? Meiosis 1, meiosis 2, like that. So prophase 1 only, what happened? All the chromosomes are arranged in a linear line in a prophase 1 of the meiosis, which usually result in sterility. Autoploidy can arise through the non-disjunction or in mitosis or meiosis. Many angiosperms are autopolyploidy. They have given as an examples. One is called as grapes, already told you. Banana, sugar beet, maize already. Rice, gram, tomato, watermelon, apple, coffee, peanuts, oats, beets and beans. Beersins. Enotarana, Lamarckiana, chrysanthemum, etc. These are all are examples of the autoploidy. Just note it down, my dear students. What is my autoploidy? How it will happen? How the chromosome of division takes place? How it will be united? They are nature or artificial. Okay. So where it happens, how it happens, which phase is the it will occurs for phase 1. Okay. What are the examples? This is very, very important, my dear students. Okay. So we will finish it up to this level only. The remaining things we will see the next classes. Okay. Allopolyploidy. Yesterday I asked the one question. Anybody else? You have to prepare and come. Ask Dino last. Any ideas? Anybody follow it? Okay. Anyhow, I summarize all those things. Okay. Already we discussed what are the things is there now. What is my ploidy? Before that, when we will study the which one you know? Hemophilia, cystic fibrosis, and the last one is called as what is that? Okay. The ploidy. Ploidy already we know, but what is ploidy? All those things. What are the types of ploidy is there? And haploidy, diploidy, and triploidy. Ploidy. Last one you must note it down. And tetraploidy. All those things. Chromosomal disorder. Okay, they may ask the questions. Which phase? Here the telophase. There. Which one you know? For phase 1 in meiosis. Note it down, my dear. How many set of chromosomes is there? Which one is sex chromosomes? How many pair is there? Okay. And what are the types is there? What is my monoploidy, diploidy, polyploidy? Here the question is that trisomic. They ask the question 2n plus 1 trisomic. Given examples. So 47 it comes, isn't it? 47 means it's also called as Turner syndrome. We'll study that things also in later. Okay. Tetrasomy 2n plus 2. Nullisomic 0. 2n minus 2, like that. These things also already we discussed. Okay, ploidy, already we discussed these things also. How it will be divided, all those things already we see also it. In which phase already we see it, in which stage the cell division is failed to during the polyploidy, telophase. And then aneuploidy, already 2 heteroploidy. What is my aneuploidy? What is my heteroploidy? It is nothing but the extra chromosome than the normal genome, number of the species. It is called as hyperploidy. How they are classified, you know, the monosomy, already we discussed the monosomy, nullisomy, trisomy, and tetrasomy. 47 chromosome it comes during the, what is that? Gamete formation 23 plus 24. The 23 combined with the another molecule, another gamete of the male or female, you will get 47. Suppose less means what happens? Suppose excess means 48. Normal 46 only. Okay. So likewise, plant as well as animals also, what happened in naturally occurs, that is called as autopolyploidy. Suppose if it is artificially we did, you know, that condition is called as what is that? Allopolyploidy. Okay, 2 plants, 2 specimens they were taken, and also hybrid itself. As a result, we will get the new one. Okay, so already you know about that things also already we discussed. So these are the things already. Double monosomy. You must note it down, my dear student. Here you have to take a examples you have to take from. What is my monosomy? 2n minus 1. Given examples. Okay, monosomy example. Later it comes, but before you should note it down. What is my double monosomy? Nullisomy. Given example. Trisomy, tetrasomy. Okay, that things also. Okay, that's all about, my dear students. We will finish it up to this level only. We will see the remaining things on the next classes. Thank you. Thank you, my dear students. Thank you, Ba. Thank you, Varsa.
Thank you, sir.
Thank you, Ba. Thank you. Take care. Bye.